First, I would like to thank the Royal College of Pathology and the Ghana College of Physicians and Surgeons for this invitation. I am Alejandro Madrigal. I am Professor of Hematology at University College London, and I am also Honorary Consultant at the Royal Free Hospital. I also have some appointments in Europe, and I will be presenting some of the work I am doing with them. I have been past president of the European Bone Marrow Transplantation. I have been the scientific director of the Anthony Nolan Research Institute. I founded the institute, and I founded also the first bank of cell therapy in the Anthony Nolan. So the work I will be presenting in some ways summarize these activities during the last years, and also where the future goes, which is very exciting, as you all know. So the talk that you had asked me to present is on human leukocyte antigens and their application, which is a very interesting and challenging topic and has been very close to my heart and what we have been doing. Let's start a little bit basic. The immune system, as you know, is the ultimate lines of defense, and you have the innate immune response that is obviously represented as a first line of defense with phagocytes, natural killers, all the cells that has capacity to phagocytes, but natural killers by themselves capable to recognize tumors. And I will talk at the end how we are actually using these cells in an innovative way to treat cancer. But the second line of defense obviously comes from the T cells that are capable to recognize specifically targets, tumor antigens and viral antigens, and B cells capable to produce, as you know, antibodies. All of them really play an important role, and our body every day is producing millions or billions of cells, like red cells. They are producing obviously granulocytes that forms the innate immune response, capable to recognize phagocytes, but also capable to present antigens, and obviously the memory cells, the T cells and B cells. And they play a critical role every day in our lives, in tumor defense, in immune protection. Probably even we don't know how much many of us have been exposed to potential tumors, and they are actually capable to recognize. The recognition and role that these cells plays in the everyday elimination of viral infected cells and normal cells and potentially oncogenic or already and malignant cells is quite amazing. These cells, as you know, might be transformed. They might produce abnormal peptides that are actually processed by dendritic cells, so antigen-presenting cells, can be B cells, and they are transformed to the lymphatics where they are actually in contact with the T cells that recognize now or are primed to recognize through a mechanism that is quite complex, interacting with the B cells, T cells, dendritic cells, and then it leads to expansion of the cells that are actually capable to infiltrate the tumor, recognize now particular peptides presented in the context of HLA molecules that also will expand, and other adhesion molecules that leads to the tumor elimination operation. We could imagine almost the lymphocytes every day traveling, recognizing abnormal cells. As we are sitting here, very likely this is happening. Our lymphocytes are in some ways sort of touching every cell, recognizing anything that is abnormal, anything that comes inflamed or present different peptides are actually activating the T cells that leads to cell interaction in a close contact that leads to cell destruction, as you see here, in a dramatic ways, to using different ways, first, perforin and other substances that make the cells really damaged. They are also capable to release cytokines that attract other cells. So the whole process leads to inflammation, but also elimination. And we have seen in COVID how this cascade, the cytokine release storm that leads to excessive response of cells, excessive response of cytokines can lead to massive inflammation beyond what we would expect. But controlling the immune system is quite a challenge, even by their own cells. So how this interaction, how these cells are capable to interact and these lymphocytes and recognize what is going on in the abnormal cells. If we will look in detail, this slide that I just presented, we will see this. We will see a T cell, a lymphocyte, recognizing an antigen-presenting cell that now is telling that a T cell that there is something abnormal. through the HLA molecules. As you know, they are codominantly expressed. They come in different flavors, HLA A, B, and C for class 1, DR, DQ, and DP for class 2. Obviously, there are multiple molecules in the surface of the cell. You could expect that about 250,000 of each allele. So you will have 250,000 in these cells of A3, 250,000 of A1, B8. And we know that one particular antigen-presenting cells, in fact, one HLA molecule along with one peptide, is capable to activate T cells. Normally, there are more. There are 10,000 or 50,000 molecules of the same allele presenting the abnormal peptide, a viral peptide, a mutated peptide. On the other hand, as we know, the T cells will have the same receptor and is specific only for one particular peptide and for one particular HLA molecule. So this T cell will, for instance, will recognize only the peptide of cytomegalovirus in the context of A1, but won't recognize the B8 peptide unless there is a cross-reactivity. And there is always a chance of T cells having cross-reactivity, and that's why sometimes we see autoimmune disorders and other complex diseases that are recognition, or we have the capacity by this cross-reactivity to broaden our spectrum of potential tumor antigens, because we tend to deny T cells in the periphery and in the memory, we wouldn't be able to respond to all the challenges that normally are present during the life of an individual. These molecules that we call HLA molecules, I have been studying them for several years. And I would say I am in love with them. They are beautiful molecules. You could imagine this as a sculpture. They are the most polymorphic protein in our bodies, the most polymorphic diversity of genes and alleles within the same individual. All the cells will express the same set of HLA, like a barcode, I will say. All the cells will have, of an individual A, will have the same barcode. Regardless of where you take the cell, all of them will have the same barcode. How they were identified? Well, the story is fascinating, and I could spend hours talking about this, in particular because I had the chance to meet many of these people, even working with some of the people who were capable to do that. Peter Guerrero I didn't meet, but as you know, an English guy who was studying how animals reject tumors, and he identified for the first time what he called the HLA, or histocompatibility antigen 2. He unfortunately interrupted his work because the war came after in 39s and 40s, and he stopped his work. He was associated to as a military, but at the same time, Sir Peter Medawar, who would get the Nobel Prize, an Englishman, also was trying to understand how he could transplant patients with the skin when he was looking patients that were subject of a burn or damage during the war. And he carried animal experiments, fantastic and the work that led him to understanding the mechanism of T cell recognition and rejection, and for that he would get the Nobel Prize several years ago. Meanwhile, John Snell in the United States, in Ann Arbor, was looking animals that they were capable to make sort of the same strength of animals. So when the war finished, Peter Guerrero moved to the United States and worked with George Snell, and they published important papers based on the observations originally from Peter Guerrero. Unfortunately, as you see here, Peter died in 1961, and Snell would receive the Nobel Prize in 1980, together with Jean Dosset and John Baruch Benacerraf. Many people probably couldn't get this Nobel Prize because, as you see here, in 1958, in different countries, in Holland, Professor John Van Root, Professor Jean Dosset in Paris, and Ross Payne in the United States were capable to identify, recognize for the first time human HLA molecules. And John Dosset was the only one who would get it, but it's very questionable, and still, I think there are people who believe that other people should get the Nobel Prize. But for instance, I had the opportunity to work with Professor John Van Root, an extraordinary man. I was his secretary during the presidency of EBMT. I was his secretary during the chairman of, he had an immunology working party. But he was, as I say, an extraordinary man, discovered the HLA, founded the European Bone Marrow Transplantation, founded World Marrow Donor Association, that I will talk a little bit about that, Eurodonor, and was very much involved in the understanding of non-inherited maternal antigens and inherited paternal antigens. He, even until his last days of his life, he was actually very active. He died actually the other way around. He died in 4 years ago, 5 years ago, so this date is wrong. But Yando said, as I say, was actually also a gentleman. I had the opportunity to work with them. I actually did my PhD with Lady and Walter Bodmer, who were actually with Ross Payne and were contributors to this work that was published. And obviously, when I was at Stanford, I had the opportunity to work also with Ross Payne. And she was by then retired, but she would come to the sessions, and she was always so clever. There were other people who make substantial contributions in understanding the diversity of HLA and also how we recognize these molecules. We have here Fritz Bach, who discovered or invented the mixed lymphocyte culture, with Professor Edmund Yunis, who was my mentor when I was in Harvard, Baruch Benacerraf, as I say, got the Nobel Prize with Dosset and Snell, and Paul Terasaki, who also worked in England, and he developed the methods, so Terasaki trace, we call it, to detect initially the diversity of HLA. And then there was the people like Sinkernagel and Peter Doherty that would get the Nobel Prize also for understanding the interaction between the T cells, the peptide in the context, and Jack Strominger and Don Wallis with Pamela, who did the crystallography and helped us understand the structure of the HLA molecule. The structure of the HLA molecule is quite interesting. It has been actually evolving from immunoglobulins and other products through different species. But as you see here, in the case of class 2, they are 2 different chains with the alpha and beta and different domain, beta 1 and beta 2. In the case of class 1, is one single domain associated to beta 2 microglobulin and with a group where the peptide comes with a transmembrane and intracytoplasmic region. If we look at the molecules, for instance, HLA class 1A, for instance, from the top, this is a crystallographic structure which shows a group on the top here where the peptide is presented. It has 2 alpha helix with a 3.4 pitch or rotation of amino acids. So these amino acids, as you see here in different colors, have the capacity to interact or not with a potential peptide, and by doing that, capable to present. So you see here the top of the molecule, and you see what we call it pockets, where the actual peptide sits. So you have an HLA class 1, approximately 9 amino acids peptides, fragments from virals, from tumor antigens that are going to fit or not within the HLA molecule. So how this molecule and HLA works, as you know, a virus gets infected in the cell. Imagine that this is the cell, the virus is getting in. The cover of the virus, let's say the protein, for instance, Cytomegalovirus, the cover, the protein PP65, which is like an envelope of the DNA, it is fragmented by proteasomes in small peptides that are transported to the ER, where they are associated then with the heavy chains of HLA and the beta 2 microglobulin, forming this integrated protein that is then transported to the surface, and the cell is then capable to present to the T cells a foreign peptide that initiate the cascade. As I say, HLA is the most polymorphic system that exists. It located the genes in the chromosome 6. It's one of the most dense regions of the genome. There are 224 genes identified in a very small region, 3.6 megabytes, an extended cover of 7.6 megabytes. As you see here, 40 of those genes have functional with the immune system, which is quite interesting, and they obviously play an important role in defending and protecting the individual. As I mentioned, very polymorphic. I made this slide several years ago, in March 2006. At that time, the number of HLA molecules, or genes that we knew, was limited. It was much better than when I started the field in '83. But here you see that there were, at that time in 2006, only 485 different subtypes of HLA, a double number for HLA-B, and a quarter of number for, or half of number for HLA-C, and similar. Just keep these numbers in mind, 485, 816, this is 2006. See what happened just yesterday. I made this slide taking the information from the database that is produced at the Anthony Nolan by Professor Steve March, who used to work with me. So there are over 7,000 HLA. 7,000 in contrast, let me go back, 885. So the number of alleles that we are identifying different is enormous. And you assume that an individual will inherit a paternal and a maternal subtype of these 7,300 and similar for other ones, the combination that you can generate with all these individuals, creating the barcode of the individual, one paternal and one maternal for each locus, A, B, and C. So you have 12 bars. You will have an enormous diversity that in some ways protects us, brought the capacity to present peptides, as I mentioned and recognized, but on the other hand, has the complexity that in some ways transplanting or using them for matching has been one of the major problems. Just to give you an idea, I am showing you here two subtypes of HLA B35. B35, there are many subtypes, B3501 and B3503, differ by one single amino acid. In the bottom of the group, you see here the alpha helix that I already speak, the beta strands, and you see that there is only one single amino acid difference between these two subtypes, a serine from a phenylalanine in this strand in position 116. How that can check and affect the peptide binding? I mentioned that these amino acids interact with the HLA with the peptide and allow the peptide to fit or not. Let me illustrate that clearly. So you see here how this phenylalanine has actually affect. So the difference, remember, serina for phenylalanine. So you see here how the tyrosines, in the case of B3501, because there is a phenylalanine, it fits nicely here, and it gets a very nice association. Whereas in this case, this serine, phenylalanine, impact is strong. It doesn't allow the peptide to bind. So that could affect the structure of the molecule, the fitting of the peptide, and that would be the mechanism why it won't be normal recognized as a cell, and then that would trigger the T-cell response that I mentioned, and I show you so clearly that triggers cell destruction. So one single difference between, for instance, a donor, imagine that this is a patient that has B3501, and this is a donor, and we type in wrongly, and we transplant the patient with one single amino acid difference. Now this individual will have all the cells of the immune system of the donor recognizing that there is something wrong, and it will lead to T-cell destruction and elimination of the cells. How this polymorphism has been generated? How is the most polymorphic gene? We at one point looking the polymorphism at that time in 2000 in 2000, we believe that represent 15 or 20% of novel alleles reported by recombination, where there is a recombination of certain elements of the HLA molecules that generate other elements. Now, we know that many of them are due to point mutations. Just to illustrate that, this slide is also produced at Anthony Nolan by the informatic group. You see here the different exons, so the leader, the alpha-1 domain that I mentioned, alpha-2, and so on. And these are the amino acids located in each area, the alpha-1 and alpha-2, obviously that are more related, where the peptide, the alpha helix, as I show you, the beta strand, this is the transmembrane and intracellular region. So you see across the polymorphism, as you expect, is very much related with the areas that are interacting with the peptides. So in this column, it means that in this position, there is a possibility to have several options of different amino acids. But now look what happened in 2016 with all the new molecules that we have identified. If we put all the variations that are in HLA-B, we see that the polymorphism has the HLA polymorphism occur even in intracellular regions that might affect even the structure that leads to present or even bind to the membrane. So one of the challenges with this enormous polymorphism is how we detect all these variations. Initially, as I mentioned, thanks to Terasaki, we were using antibodies to type these individuals. When the time came in the use of DNA, we were capable to develop structures and methods that help us to recognize using oligoprobing systems, using SSO, SSOP, or SSP, and later on sequence-based typing. In fact, this has been a major challenge in the 80s, 1980s, it was only serology. With the starting to use DNA, we started to use in the 90s the use of RFLP, and I remember the times where we used to do very complex ways to recognize these polymorphisms, and also impossible to put them practically in routine. So then the use of sequence-specific priming allows to identify variabilities on this. And then we invented a method that we patented in 1986 that was called reference transconformation analysis that recognized single nucleotide variations, and at one point was the most useful method to recognize the polymorphisms. But then it was superseded by sequencing. So we see as time went by that by 2003, 2000, we started to use Luminex that use a commercial tools that help us to recognize this diversity using, again, methods like SSO and SSP, but then it came the methods for typing and sequencing, in addition to the Sanger methods that were typing. And then we started to use the third generation sequencing and the next generation sequencing. So thanks to that, now we can actually analyze practically every intron and exon of the region, as I will show you, and understand the impact that this have. Also has made the nomenclature a little bit more complex. So before, when we use serology, we call it only HLA-24. Then we identify other polymorphisms, and there was A2402, 2404, whatever. And then we have different subtypes that recognize silent substitutions, and even the expression, as I mentioned, the modifications can be in the intracellular, could lead to low number expression or null, or it could be secreted because it doesn't allow to anchor, and questionable is when it's not clear. So the challenge has been to introduce every time new ways to do that. And obviously, at the moment, we use next generation sequence that are available, and we have been working with industry. At the Anthony Nolan, we developed the third generation sequencing, and I will show you what we call ultra sequencing. And that helps us to type to the allelic level. So this is a report of several donors. You see 1, 2, 3, or whatever, and you have HLA, and you see how many digits you can actually have. So we have all the different subtypes. In the past, we only used to call A2, and probably this patient was, or this donor was very different from what was another one that has a different subtype that we even didn't know. In fact, for A2, which is the most common HLA allele in the population, there are over 500 different subtypes. It's incredible to imagine that one single type of HLA serologically recognized has about 500 different subtypes because it has different variations along the whole gene. So this has been one of the challenges during the years, matching and typing. Another challenge has been to understanding the contribution of each loci. I mentioned that each individual will express two codominantly. In this case, the father or mother of this individual was A1, A24, and similarly for the other alleles. So finding a perfect match and understanding what contribution, for instance, what happens if we mismatch HLA-DP, or what happens if we match only, which is more important, should we mismatch if we have only a difference, if we have only a donor available and there is no one else, or we have two donors, let's call it like that, and one has a difference in the HLA-B and one has a difference in the HLA-A, which we should use. And the truth is that we don't have that clear, despite so many years. It still is not very clear the contribution of a mismatch in the outcome of the transplantation. Transplantation, stem cell transplantation, is again something that we could remote to the early 50s with Donald Thomas and other people doing animal studies, trying to understand how they reconstitute animals that were radiated or irradiated after the accidents that happened in Yugoslavia, for instance, with reactors, as we are afraid now, or what happened, unfortunately, in Ukraine. But what happened at that time was the outcome of these patients were practically very bad. They were irradiated and they couldn't actually reconstitute their cells. So the idea was to replace their cells, rescue them with a graft of a donor. But because HLA matching was not very clear at that time, the level of rejection and death of the patient was terrible. So it was really until people started to use first using related donors, and that was extremely successful. And in the 70s, something happened very interesting that I will come back. But you see here how, through the history of this, we have been using to treat patients with sickle-cell anemia, with other conditions, even not just cancer, but also what we call minority or rare disorders, as we see here, thalassemia, and we see these other conditions. And obviously the numbers have been expanded, and I will talk about this difference. And it could be autologous or could be allogenic. And also we have been involving in making other methods that help to introduce the transplant. But I was saying that in the early 70s, in 1974, something happened that made transplantation more successful and more available, let's call it like that. The story goes that this lady called Shirley Nolan, having an Anthony that was born with a minority rare disease with Con-Aldrich in 1971, she realized that there were the first transplants mainly related because there were no registries, there were nothing. So she came to England, although she was English, she was living in Australia, she came, and she was told that there was no chance for Anthony. The only chance was to find an available and related donor that was identical to Anthony, but there were no registries. So she said, well, how I can do it? She said, well, you will have to find 100,000 or 200,000 people to find someone who potentially could match Anthony. He said, well, if that's what is needed, I will do it. And she went to the pubs, to the schools, to the churches, to the fireworks station. Amazing, amazing. She demonstrated outside the house of the Prime Minister. She was even detained by Scotland Yard because it created so much convolution. But she had the power of conviction. The first donors that actually became enrolled were the bobbies, the policemen, the British policemen that were actually taking her. And Anthony unfortunately died, as you see here, in 79. But by then, the registry had 50,000 donors, an amazing number. And thanks to her inspiration, the number of donors now in the world is incredible. More than 39 million in 56 countries are actually enrolled. They have, let's say, give a little bit of blood. We have their barcodes in the World Marrow Donor Association, that, as I mentioned, was developed by John Van Root, John Goldman, and Donald Thomas in the 1970s. And thanks to that, they hold a registry of all the donors that are around the world in these 56 countries. And thanks to that, also they have cord bloods, and thanks to that, over 20,000 patients have received last year just donors, but over 1.5 million, if no more, donors had been saved because there were donors available capable to give their give stem cells, risking potentially their lives to save the life of somebody else. So this is an amazing story. And one of the challenges has been to type these donors to the allelic level, and you see that nowadays with the new technology, a large number of them are fully typed for HLA-A, B, and C, and even for some DP. Initially, it was only basic typing, practically serology, but that has changed now thanks to the new technology. And that obviously has helped, as I say, to provide more donors. This is just what happened, for instance, in 2019, that is the last report of World Marrow Donor Association, where 22,000 were produced. And we see a little bit of decrease by the impact of actually COVID, where actually affect the borders, the availability of hospital service, and other associations. And we obviously see that was also impacted because we can provide bone marrow, but also hematopoietic stem cells, mobilized stem cells that are important for doing that. The capacity with these cells to treat patients with terminal diseases, malignant disorders, but also metabolic and also genetic disorders is amazing. You see here that the main indication in adults in Europe was mainly AML, leukemias, and we have also ALL, but also we see other conditions. We see lymphoma, we see other conditions, even autoimmune disorders or other conditions. In pediatric, things change. ALL is the most common, and PID also is important. But we have a large gamma of treatments, solid tumors, non-Hodgkin lymphoma, and other conditions that can be actually treated with the stem cell transplantation. So there are different sources where we can obtain the stem cells and the donors available. The most common donor obviously should be the identical sibling that, as you expect, has a similar chances, and because now we can transplant older people with different conditions, we see a little increase of that. But the unrelated donors in red here, you see how it has been growing during the years with the more available donors through different registries and different typing. But what it has been also very important is to use unrelated type mismatch haploidentical transplant that has increased in the last years because we can use unrelated donors, related donors per mismatch by using different protocols that allow us to eliminate the actual post-transplant reactive T cells by using cyclophosphamide and other reagents that help us to eliminate the alloreactive T cells at one point. So still matching is quite important, and not only matching for HLA, but potentially providing the donor that is compatible for many other options. For instance, younger donors, we know that they have better chances to survive. Patients have better chances to survive, at least 10%. Male donor seems to be better than any other female donor. And all that has changed the landscape of the registries. And you see that also matching for cytomegalovirus and HLA is also critical in terms of improving the outcome. That can go as low as 30% and as high as potentially 60%. In fact, using the third generation sequencing, we have published recently data that shows that matching for HLA to using the ultra high resolution, that's third generation sequencing that type the whole gene, including the intronic regions, we see a substantial improvement. We also match for cytomegalovirus, and also we take into account potentially the role of T cell mismatches. And you see these patients that are mismatched, 10 of 10, that means that 2 alleles or 3 alleles were mismatched. Well, it could be the Q and the P. And you see that there were mismatch for cytomegalovirus. In this case, this patient probably should not be either transplanted or should receive another source of the cells like cord blood or haploidentical cells. So in general, we always say that approximately, unfortunately, after the transplant, after the difficulties in finding a donor, still we have a lot to do. 50% of the patients post-transplant can die, and they have complications of death or relapse, the disease come back, although we believe, as I will show you, that the T cells will play an important role, 20% because it produces graft versus host disease, and also because infections plays a role there. So there are non-related, non-relapse-related mortality and related relapse mortality. So ideally, what we have been aiming for many years is to take cells of unrelated donors, not the whole graft, but take cells that have been trained, let's call it in the laboratory, T cells that has been expanded to recognize specific targets like cytomegalovirus. So if the patient has infection, we can just take off shell a pool of T lymphocytes that could be used to treat a patient with cytomegalovirus or Epstein-Barr or other conditions, that we would be able to remove the T cells that are actually capable to recognize graft versus host disease, but also enlarge the number of cells that are capable to recognize the malignant cells. We know that it's the transplant that actually eliminate the patient's tumor cells. We are now in a unique position. I think now, never before, we have so many tools that are going to change the way we do what is called regenerative medicine or advanced therapy. Hopefully, we might be able to limit the amount of damage that we induce with the transplant, with the chemotherapy, with the radiation, by using specific cells that have been modified to target tumor cells. So we have the cell therapy traditional, which goes from blood transfusion, hematopoietic transplantation, cord blood, and the attempt to use a specific subpopulation of cells that has been isolated, autologous or inclusive allogenic. And then we have, obviously, tissue engineering that I'm not talking too much about that today. But what has been a revolution is in gene therapy. So this, we have the capacity now to editing genes, to modify them by using techniques that are like CRISPR talent, zinc firmers. We also have the capacity to introduce more genes into the cells, ones that we modify. And thanks to the combination of these modifications in gene therapy and what happened in cells that are every day capable to be separated and isolated, we have now what we call gene-modified cell therapy that has what we call CAR T cells, and I will mention T cells that are activated or expanded, specifically recognizing tumors or viral antigens, tumor-infiltrated lymphocytes, which are cells that are actually around the tumor but have been switched off by the tumor, but we can reactivate them and induce, and also capacity to induce antigen-presenting cells and inducing. CRISP, as I mentioned, and gene therapy modifications has been an outstanding novel techniques that has helped us to correct the genes and potentially to modify. Obviously, we have to use this in somatic cells, non-germinal cells, but the process even is so simple now. You can even buy a kit commercially that has what we call the guide molecule to help you to enrich the region that you want to modify or cut, the cutting element that allow you to remove the ones that you don't want or you want to repair, and the healthy DNA strand that attach. And this is helping us now to modify many things. Another important element that is helping us is to introduce genes to these cells, to modify them. Mainly what we do is somatic cells that we modify external to the patient and we put them back with some modifications like the CAR T cells. And for that, we use viral vectors. We have now adenovirus, lentivirus, retrovirus. We use the same principle as virus infect a cell. As you know, the virus has a cover or an element of genes that help to find a receptor, to transit through the cells, and to deposit the DNA or the RNA into the cells. If we use the same knowledge and the same concept, we could pack the gene that we want to introduce into the cell using the same kind of structures of genes that, or elements, viral elements that the virus use. And that's what is coming, and this is becoming almost also commercial. So we have vectors, adenovirus, lentivirus, and other ones that help us. And thanks to that, now we are exploring and treating the patients with a large several conditions, mainly for genetic therapies, but also for cancer. So we have already approved by the EMA several elements for certain conditions. You see here that goes from genetic therapies for rare diseases, as you see here for ADA, but also for cancer, you see for melanoma, for leukemia, for other elements, for thalassemia, and all of them, as I mentioned, use lentivirus, retrovirus, and other elements that are here, and as this is the approbation. So they are already available to treat patients. So this is combining cell therapy, gene therapy, and T cells. That is an amazing achievement. One of the most novel and most revolutionary treatment that is changing the way we treat patients with leukemia and potentially will have other impacts in other conditions is what we call CAR therapy. What is CAR therapy? It's at the same time cell therapy, gene therapy, and immunotherapy. What's happened? Imagine that this patient had leukemia. What we assume for what I just told you is that the cells of the patient, the lymphocytes, have failed to recognize the tumor cell. So at that stage, there is not target recognition by the T cells on peptides presented by their own MHC. So this tumor keeps progressing and there is no way to eliminate. So the patient, in principle, if we don't do anything, will die. So we can transplant it and produce, bring in a graft that has the risks that I mentioned, graft versus host disease and other complications. Or what people has been trying, which is innovative, is trying to modify the lymphocytes and put them back to recognize the tumor. What is happening? So you take the cells from the patient, the T cells, you isolate them, and you actually load them using these viral vectors, these transporters that I mentioned, these carriers, and you load it with a lethal weapon, an antibody now that will recognize tumor cells. In this case, the majority of the CARs are recognizing CD19. CD19 is a normal protein expressed on the B cells, not just the tumor cells. but because the leukemic cells in certain types of lymphoma and ALL leukemias or the B-cell-derived leukemias, it's more likely that this presentation of this CD19 could be a target. So you actually load an antibody associated to a lethal weapon, let's call it, and then you transfect that one or put that one into the cells using this viral vector, lentivirus or retrovirus. So this going to the nucleus now made the gene to these cells to express CD19 on the surface. Before didn't express, sorry, not CD19, anti-CD19 with the lethal weapon. And then we expand these cells, and then we be sure that everything is okay, and we bring it back to the patient and infuse. This process can take up to, as you see here, up to 20 days. And actually has certain inconvenience. It's autologous, so it has to be made for every patient. It is time consuming. It takes, as I say, 20 days, and by then sometimes some patients might go into different condition. You need to isolate cells from the patient, and sometimes you don't have enough cells from the patient because already has some malignance or has been treated severely. And what is also very critical is very expensive. The treatment of CAR T cells goes in the order because the industry, pharmaceutical industry, is now helping to produce this in large scale or processing this in large scale, let's say. It can cost up to $350,000, $400,000, which is something that only some countries or some patients can afford. So we need to go around that. So people are trying to do allogenic expansion of these T cells. And obviously you can get the autologous that I mentioned that you can actually put it back to the patient as I just described. But ideally, you could use one of donors that potentially could be similar match, but the problem still is that it's a very expensive procedure. You can use T cells, but you can use NK cells. NK cells has the capacity to express, to be less restricted to the HLA requirements that I mentioned to the NK, so to the T cells, and they are also capable to eliminate tumor cells by themselves. So, but if you put this lethal weapon into that, you can produce NK cells that now is called CAR NK cells rather than CAR T cells. You can also get cord blood from different sources, from cord blood expanded, and there is a paper already published by Katie Rasvani recently in MD Anderson that shows that she can manage to get expanded NK cells from cord blood that are actually transformed and make CAR, as I mentioned, by introducing the vectors that express CD19 plus other cytokines like IL-15 and suicide genes. And she has treated 11 patients with very high success. But it's still complex because the sources of cells can change, and it's not an easy procedure. And what is attractive is what we call induced pluripotent cells. So this is cells that can be transformed into a way that these cells can be able to produce in large scale cells that we want. And that's what we have been betting. So normally normally the embryonic cells, as you know, comes from morula, blastocytes, and other lines that leads to the pluripotency. And these cells can differentiate the mesoderm that are capable to produce red cells and cardiomyocytes, or endoderm that differentiate into other elements, or ectoderm, neurons and skin. But the same now is possible thanks to the Yamanaka work. Yamanaka got the Nobel Prize, a Japanese fellow who understands that by adding these 4 genes that you see here, this cocktail that now is called the Yamanaka cocktail, these cells, normal cells, fibroblasts or somatic cells, can be transformed into induced pluripotent stem cells that are similar. In fact, they could be, for no reason, capable to produce a whole human individual, except that does not allow it. And normally the modifications in somatic cells, no germinal cells, but also at one point is mainly for using, and is highly regulated in differentiating and trying to produce these tissues. So we have been trying to use these cells, and what we have been doing is taking the cells from a bank of cord blood that we identify, that we search the most common haplotypes. And remember, the HLA I mentioned to you, and some individuals, we have a unique barcode, and some of them has a common barcode in the population. So we search for those barcodes that are common, identify the most common ones in Europe, in Spain and Europe, and then we search for individuals that will have the same HLA from the father and from the mother. So you won't have 12, because one father and one mother. So you have practically, although you have 12 in principle, but if the father and the mother has the same type, then it's 6, because you have the same subtype for each one. So this is a project that has been led by Dr. Sergio Querol, with the leader of Dr. Belen, and we have been working in doing this project. The process is simple. Once that you identify the cells, that was a major task. Once that you have these cells, you isolate the CD34 cells, the stem cells. CD34 is a protein expressed in stem cells. So you'll isolate, and normally these cells are stored already for transplant for a patient receiving a cord blood bank. So we identify 7 cord bloods that has these unique HLAs, and we add the Yamanaka cocktail and what we call the day 0. You cultivate them in different mediums, and you generate actual embryonic cells, and now you can differentiate. You see here cells that can now be lead to produce mesoderm, the cells that I mentioned that can produce the cells that we want, Tcells and other red blood cells. And you see that you can produce endoderm. The other lines that I mentioned produce other tissues, and also ectoderm. So these cells are checked, and they are sure that you have a good phenotype. So you remember, coming from cord blood, homozygous CD34 cells, you modify them with the Yamanaka. Now you have a line of cells called iPS haplobanks that could be used. And these cells, as you see here, thanks to the work of Belen Alvarez and Dr. Sergio Querol, can be differentiated in all the beautiful cells. This is like a library. You see granulocyte, melanocyte, so you can produce all lines. And we are working just to finish in a project that we hope that we will generate allogenic T cells that are actually capable to regenerate banks, and it will be off-shell using all the things that I mentioned to you. So you have iPS cells from the bank. We have ways to introduce cells using vectors, but we also have a novel system that introduce, rather than viral vectors, is using transposomes. So by doing that, we can load these cells with different genes, CAR T cells, CAR genes, suicide genes, and other elements. We have ways, as I mentioned, to differentiate it in NK cells or T cells or mesenchymal cells, and we can actually generate banks of each ones that can be off-shell at one point once they are validated and tested, being able to use and to treat patients specifically. So all these talks that I mentioned in terms of identifying cells, identifying selection, expanding the cells, being the product that is reliable, will lead to what we are seeing, a revolution in cell therapy, in the manipulation of T cells and the use of all this knowledge that I had come from the times of understanding the diversity, the HLA, the complexity of matching, the expansion of T cells will help us, what I believe, very soon, to have practically the old pharmacy, but with the cell therapy, where we have cells that have been modified to mesenchymal cells, to T cells, to NK cells, and could be used to treat particular conditions, because the target, the CAR, the lethal weapon I mentioned, might recognize some particular tumors. Other ones will recognize other tumors. Some of them might recognize even viral or fungal peptides that could lead to treated fungal infections. And some of these cells could be differentiated to why not, there is a group already working in the United States making beta cells capable to produce insulin. So with this, I would like to thank again the college. I would like to thank the college also of Ghana, and congratulate you for all the effort that you are doing, and I'm grateful for having listened to me. Thank you.